Hi. Today we're going to break some rules. We're going to talk about when genes don't segregate independently. We're going to explore the linkage of genes on chromosomes and how this can be used to map genes. Here's the voiceover. Today we're going to talk about the linkage of genes. And here I'm showing you the genetic map of Drosophila, the fly that we've discussed so much. And here you have 4 linkage groups. A number of physical traits, genes that can cause visual phenotypes that are easy to score, have been mapped to 1, 2, 3, or 4, one of these linkage groups. And linkage group 1, of course, is the X. So today I'm going to explain how this was done and what this says about how genes both segregate together and how genes can recombine in meiosis. So these are deviations, in a sense, from Mendel's laws of inheritance. And to remind you of those laws, Mendel's first law stated that each individual organism possesses 2 alleles that can code for a trait, and these alleles segregate when gametes are formed, and 1 allele goes into each gamete. Mendel's second law states that these different alleles at different loci separate independently of one another when the gametes are formed. This is due to the independent separation of homologous pairs of chromosomes during meiosis. Let's say you have 2 flies, each heterozygous for 2 different genes, and each carries a dominant version of one of the alleles and a recessive version of one of the alleles. When you do the Punnett square for a dihybrid cross, which is what you would need to do here, these 2 genes would independently assort. These are the genotypes you would get in the next generation. And this is the phenotypic ratio you would see, 9 to 3 to 3 to 1. Now, this doesn't always occur. So Bateson, Saunders, and Punnett in 1911 noticed an apparent inconsistency. So looking at these plants here, they had pure breeding strains that contained purple flowers and produced long pollen. They also had another strain that always produced red flowers and round pollen. They crossed these together and they noticed that they got only purple flowers and long pollen, or that would be what they so that's what happened then. And so that means that purple flowers and long pollen are the dominant alleles. But something interesting happened in the F2 generation. So after these F1s were selfed, the ratio of the F2 generation was not the 9 to 3 to 3 to 1 ratio. What they got was something a little bit different. So it seemed as if in some ways these genes could have been linked. So if we look at the real data, easier sometimes to put the crosses into 16ths if you want to compare it to the 9 to 3 to 3 to 1 ratio, because that adds up to 16. You express that your real data in 16ths. You create the ratios. So for example, the number of progeny with a phenotype over the total number, and you want to get that to be equal to x over 16. So you can do some algebra and express it this way, and basically multiply what you get from the division by 16. So in the F2 generation, this is drawing upon the data from the last slide, we got these number of individuals, 284 of the purple and long pollen. So that is an 11.9 when you express it in 16ths. For the purple and round pollen and the red and long pollen, you get 0.9. And for red and round pollen, you get 2.3. So the ratio is 11.9 to 0.9 to 0.9 to 2.3. So that is not, then that's approximately 12 to 1 to 1 to 2, which is not the 9 to 3 to 3 to 1 ratio. So there must be some sort of linkage going on here. So Morgan, he explored this further. He observed when he had two genes that were tightly linked on the X chromosome, R and L, and he crossed a female that was homozygous dominant for this with a male that had the recessive alleles, he would get these genotypes in the F1 generation. So the female would have both a chromosome with both dominant alleles and a chromosome with both recessive alleles. In the F2 generation, if we cross the male and the female, you can produce these gametes. And you should get these types of progeny if these genes are really linked. The males will get a X chromosome that has the dominant version of both genes, giving them red eyes and long wings, or the recessive version of the gene, getting white eyes and short wings. What were the males that he actually found? So he did find males that had both the dominant alleles, both the recessive alleles, but then he had males that had one dominant allele and one recessive allele for both of these traits. So he could get red eyes and short wings together, which is not what you would expect, or white eyes and long wings together, which is not what you would expect from a linked gene. Linkage can be broken. And so this was something that was interesting to Morgan. He turned to some undergraduates for help. So Sturtevant and Bridges were both undergraduates in his lab. They took his genetics class, and then they joined the Morgan lab after they took the class. They were wondering, how could this apparent break in linkage happen? These genes have mixed and matched somehow, but how did they do it? So Morgan suggested that the X's were somehow intertwined, and this entwining must have caused some sort of rearrangement between the chromosomes in the female parent. Sturtevant thought it would be possible to deduce the order of genes from this process, which is a pretty amazing insight. He tried to use this phenomenon, which would be called recombination, to build a genetic map of genes on the X chromosome. And here's Sturtevant's map. There were a number of genes that were already mapped, so this is not all of his work. He took all the data that the lab had from all the breeding records that they kept, and he looked at the data from over 21,000 flies, and he found that only 214 had white eyes and yellow bodies. So what was going on? He argued that yellow must be close to the white gene. He decided to call this 1% chance of getting both of these genes on one chromosome a map unit. And he would use this recombination frequency of 1% to say that white and yellow are just one map unit apart. He also did similar crosses and looked for flies that had yellow bodies and vermilion eyes. And he found that 32% of them would become recombinant, which is a much greater number. Therefore, yellow is 32 map units away from vermilion. Then he looked for flies that had both white and vermilion eyes. And he found that that occurred about 29% of the time. And all of these things are done with crosses where these genes are not on the same chromosome. There must be some sort of exchange going on, so there has to be recombination. So he used this information to figure out that white was 29 map units away from vermilion. So how are you going to put all this together into a genetic map to figure out the position of these genes on the chromosome? So we know that yellow is one map unit apart from the white gene, and that yellow is 32 map units apart from vermilion. and white is 29 map units away from vermilion. If you're wondering what side of yellow white is on, it cannot be on the left side of yellow. It has to be between, white has to be between yellow and vermilion. Barbara McClintock and Harriet Creighton studied corn, and they had some interesting corn specimens that had some chromosomal abnormalities. And they used this to discover that intrachromosomal recombination, such as the Morgan lab was working on, requires the physical exchange of chromosomes. So how did this process work? It involves reciprocal exchange. So they could mark the chromosomes and detect abnormalities in the chromosomes visually. So they had a chromosome that had a heterochromatic knob at one end and one that had a piece of chromosome 8 fused at the other end. These abnormalities were also genetically marked. You had a dominant allele for colored kernels in the corn, and you also had a recessive allele for colorless corn kernels, and you had also a dominant allele for starchy kernels and a recessive allele for waxy kernels. You can set up crosses where you have both the colored kernels associated with the waxy kernels and the colorless kernels associated with the starchy kernels. And you can cross these to homozygous recessive corn plant. You get a chromosome that is colored and waxy, and then one that is colorless and starchy. And these chromosomes look different. And they found recombinants that were both colorless and waxy. And so you can see that the chromosome looks different. There's a rearrangement that has gone on. And also they found chromosomes that were colored and starchy. So from this idea, the linkage that people were discovering in genetics was due to the physical exchange of parts of the chromosomes. So let's talk a little bit more about linkage. So what if two alleles are on the same chromosome and they're close? These are linked genes. We can look at, for example, two genes, R and Y, and we can cross them together in the parental generation, something that's carrying the dominant allele for both and something that's carrying the recessive for both. And they should produce two types of gametes, dominant, recessive, recessive. In the F1 generation, you should produce animals that are all heterozygous for these traits. And if you cross them together, you can produce these gametes. And in the F2 generation, if you do the Punnett square, you should get these genotypes. So you get 1/4 dominant, 1/2 heterozygous for both genes, and 1/4 that are recessive for both traits. So this is almost like having just a single gene because they are so tightly linked. There's no separation between these traits. So this is how we do notations for crosses with linkage. You can see if you have a gene A and B, there's a dominant allele that's uppercase for each of them and a recessive allele that's lowercase for each of them. This is how we would write them. They would produce these gametes. And if they're tightly linked, you would get only this type of progeny where you'd have A and B, they're on one chromosome right next to each other, and you would get both versions, either the recessive or the dominant versions. A and A are never on the same chromosome, right? Because they are two alleles of the same gene. And these genes are always found in the same order. Now, how do these genes segregate? If you take homozygous parents and you cross them, you will get heterozygous parents at both loci in the F1 generation. You get very simple genotypic ratio of 1 to 2 to 1. And this is, yet again, another example of when genes are tightly linked, they basically act as if they were the same gene, even though they're two different genes. Because they're so close to each other, they segregate. And this occurs because there's no crossing over between them during meiosis. But crossing over, of course, does occur. And this crossing over can cause the production of recombinant chromosomes, where you actually, in building on the example we were using, you'd actually get a recessive allele of one of the genes and a dominant allele of the other gene together on one chromosome. That's what I'm showing here. So if crossing over does occur, you can get recombination, and I'll go through this again here. So if you cross two flies, let's say, that carry two relatively closely linked genes, we don't know how closely linked they are, but let's say there's some linkage on the same chromosome. And these are genes A and B. And you have one that's homozygous for the dominant allele, and you cross that one with one that's homozygous for both recessive alleles of these two genes. You would get these types of gametes. And in the F1 generation, if you cross them, you would get recombination occurring between these two chromosomes. So the gametes formed from the F1 cross could be recombinant, where you get a combination of dominant and recessive alleles on one chromosome, or non-recombinant, which are just like the chromosomes of the parents. So using this recombination frequency, we can learn a lot, and I'm going to tell you now how to determine it. So just like was done in Morgan's lab, recombination frequency is the percentage of recombinant progeny produced in a cross. And here we have the formula for the recombination frequency, which is the number of recombinant progeny over the total number of progeny times 100%. So in this example with plants, you get 8 and 7 recombinant progeny. This is different leaf types and also based on a height gene. And you put those over the total. So you get 15 over 123, and you get a 12% recombination rate. These two genes are 12 map units apart. To summarize the linkage of genes, we knew that genes at different loci exhibit independent assortment, but genes may be linked, especially when they are close together. In some cases, they are too close and they don't independently assort at all, so this is complete linkage. In other cases, they are linked but not so close, so crossing over can take place between the genes, and this is incomplete linkage. Individuals that are heterozygous for two linked alleles will produce two types of gametes, two non-recombinant and two recombinants, and the recombinants occur occasionally. There are two types of recombination: interchromosomal, and this occurs between genes on different chromosomes, and this happens due to independent assortment, and intrachromosomal, genes that are located on the same chromosome. So we've discussed linkage. Why don't you take a break, and then we're going to come back and dig deeper into genetic mapping and also talk about some more modern ways of doing this. See you in a minute. Drosophila melanogaster has 4 linkage groups that correspond to its 4 pairs of chromosomes, and these are depicted here. There was a lot of work done to determine the recombination between these genes to generate this map. And as you notice, all of the genes that are marked have visual phenotypes, so it was easy for the researchers to see whether a particular allele segregated together with another gene because they could simply observe the differences in how the fly looked. Gene mapping and recombination frequencies can be used to make a map like this, right? So the physical distance between genes determines the rate of recombination. Two genes that are far apart are more likely to undergo a crossover. From this, you can determine the recombination frequency, and this can be helpful to provide the order of genes and give an estimate of the relative distances between them. They cannot exceed 50% recombination. If that's the case, then the genes are not linked. They're on a different linkage group. So they're located on a different chromosome or different linkage group, as I said. The linkage of genes calculated using recombination frequencies is a genetic map. Distances are measured in map units. One map unit equals 1% recombination, which equals 1 centimorgan, and that is named after Thomas Morgan. The linkage of genes calculated by physically locating the gene on the chromosome is a physical map. In this graph, you can see that recombination frequency increases as the distance between genes increases. Gene mapping, as we've discussed, occurs using recombination frequencies. So let's use another example. If gene A is 5 map units from gene B, and gene B is 10 map units from C, and if gene A is 15 map units away from C, then A and C are the furthest apart. So you can construct this basic map. A and C are 15 map units apart in one direction, so the order here of the genes is not clear. But using the information from B, we know that B must be between A and C, but it could be in either this orientation or that orientation. Now, to figure out what the orientation is, we need another gene. So we know that A and B are 5 map units apart, and B and C are 10 map units apart. there's another gene, D, and we determined that A and D have a recombination frequency of 8%. So they're 8 map units apart. B and D are 13%. They're 13 map units apart. And C and D are 23 map units apart. The higher the recombination rate, the further they are apart. So D and C are 23 map units apart. And using the data also from B and D and A and D, you can construct a genetic map like this, where D is on the left, A and B are in the middle, and C is on the right. So D was very useful in figuring out the order of these genes in the linkage group. So you can construct a genetic map using multiple two-point crosses. Here we have A and B, A and C, and A and D all with 50% recombination. So that means that they are the furthest apart and probably on different linkage groups. And B and C, B and D, and C and D all have less than 50% recombination frequency, so they're likely to be linked. So you can put A onto linkage group 1, let's say, so it's separate from the others, which are on linkage group 2, and you can figure out their order because you know the distance between D and B, D and C, and B and C, all generated from the recombination frequency. Now, this can be done a little bit more quickly if you use 3 genes at once. If 3 genes are linked, during recombination you have 3 possibilities. A single crossover between A and B, a single crossover between B and C, or a double crossover where B is exchanged between the 2 homologous chromosomes. So let's say we have a wild-type fly and a scarlet ebony spineless homozygous mutant fly. These are 3 different mutations in Drosophila, of course. Scarlet eyes have a more reddish look than normal eyes, and then we have red eyes, which is the wild type. Ebony body color is more dark, and then the normal fly has, I guess, more of a gray look in comparison to ebony. And then you can also look at the bristles on the fly, and in spineless you have much smaller bristles than the normal bristles, which would be the wild type copy of the gene. So the mutations are all recessive traits, and we could put them together in an arbitrary order, but to figure things out, we would have to do a Testcross. And I'm going to show you a small video about how this is done.
We will use Drosophila to see how a 3-point Testcross can reveal the order of mutant loci on a fly chromosome. In the triple mutant fly, scarlet eyes are recessive to wild-type red eyes, ebony body color is recessive to gray body color, and spineless bristles are recessive to normal bristles. All 3 mutations are linked on Drosophila chromosome 3, but in what order? We set up a 3-point Testcross to examine recombination among the mutant loci. Our triple mutant male parent is homozygous for all 3 mutations. All male gametes will carry all 3 recessive mutations. No recombination can occur. Our wild-type female parent is heterozygous for all 3 mutations, and the mutations are coupled on 1 chromosome. Meiotic recombination in this parent should be easy to detect in her progeny. Let's take a look at crossing over during meiotic recombination. Recall that in a 3-point cross, 2 crossovers are needed to produce a double recombinant during meiosis, 1 on each side of the middle gene locus. As a result, double recombinants are the least common meiotic products. Non-recombinants with no crossovers are the most common products. The middle gene locus is always the one which is recombined by such double crossovers, as we see here in the gametes, and eventually in the double recombinant progeny. By doing the 3-point fly cross, we can easily learn which mutation maps between the other 2. Our Testcross produces 755 progeny, expressing 8 different phenotypes. As expected, the most common progeny are non-recombinants, wild-type heterozygotes and triple mutant homozygotes. The double recombinants, spineless flies, and scarlet-eyed ebony flies are easy to identify. They're the two least common classes of progeny flies. Spineless progeny differ from wild-type non-recombinant progeny at a single locus, spineless. Similarly, scarlet-eyed ebony progeny differ from the triple mutant non-recombinants at a single locus, again, spineless. Clearly, spineless is the double recombinant locus that maps between the other two mutations.
So let's try to put these genes on the genetic map. We know the order of these genes. We know the distance between each of one of them. And by putting that data together, we can generate a map that shows spineless in the middle, ebony on the right, and scarlet on the left. Now with this map, this tells us the order of the genes. We assume a physical distance that separates the gene, although we don't exactly know what that distance is because we haven't created a physical map, but we will discuss that later. And we have deduced this all from the proportion of recombinant and non-recombinants that can be produced. We can use this to predict that in the future. So how do we do this? Scarlet and spineless are 14.6 map units apart. That means 14.6 of the gametes would be recombinants. So if we know the recombination frequency, we can calculate the rate of double recombinants. So you have to remember the law of probabilities for two independent events to do this. So we have our genetic map. So now, how can we determine the rate of double recombinants? Scarlet and spineless are 14.6 map units apart. So there, like we just said, 14.6 of the gametes will be recombinants. Similarly, 12.2% of the gametes will be heterozygous for spineless and ebony, based upon this map. So if we know the recombination frequency of individual events, you can calculate the rate of double recombinants by multiplying them. So we have 12.2 over 100 times 14.6 over 100, which gives us 1.78%. By calculating all the recombination frequencies between these genes that have a visual phenotype, a complete linkage group for each of the chromosomes have been established in Drosophila. And through further work, we've been able to create a physical map and link this to the actual physical position on the chromosome. So how did we do that? So to make a physical map, you remember, for example, with the human chromosomes, you can create a karyotype and look at the chromosomes directly, and you can stain the chromosomes. So you can use fluorescent and situ hybridization fish to label chromosomes by using the sequences of certain genes. This probe that you make will only hybridize to the chromosomes that contain that gene. So these molecular probes are tagged and they're sequence specific. They fluoresce under UV light. You mix them with the chromosomes using a somewhat complicated preparation. And the fluorescence of the probe tells you where the gene is located. Fluorescent in situ hybridization fish has been used many times to identify the location of genes to generate a physical map. And here's an example from another fly, Drosophila bazadi. And you see this one, it has 6 chromosomes. And what was done is they hybridized large chunks of the DNA to these chromosomes, and they determined where these large probes hybridized, and that gave them the ability to create a physical map of this fly that has 6 chromosomes, X, 2, 3, 4, 5, and the small chromosome 6. Gene mapping in humans is a little more complicated and has relied upon pedigrees. The first documented case of linkage in humans involves the pedigree analysis of ABO blood type and the nail patella syndrome, which is an autosomal dominant disorder. This causes abnormal fingernails and an absence of rudimentary kneecaps. This syndrome is relatively rare. Affected individuals are heterozygous, obviously, because it's an autosomal dominant disorder. Unaffected people are homozygous. What was interesting about this case is that there was a linkage between ABO blood types and the nail patella syndrome. So if you look at this pedigree, the father in generation 1 has nail patella syndrome, and he is of the A blood type group. He is actually AO. And the mother, she is of the B blood type group, and she's actually BO. In the father, the autosomal dominant gene for nail patella syndrome is linked with the O allele. Okay, so you can see that. In the second generation, all children with nail patella syndrome have either blood type B or blood type O, indicated that the genes for the syndrome and for the blood type are linked. Nail patella syndrome does not develop in children of generation 2 with blood type A, right? Because that blood group is not linked with the disease allele. In generation 3, we get a child with a type O that has no nail patella syndrome and a type A with no nail patella syndrome. These outcomes are the results from crossover events, because if you look at the parents of both of these in generation 2, you can see that the affected father, in that case, he had that linked to the O allele, which is represented by the small I. The linkage had to be disrupted, and that's an example of recombination. The same goes for child 3. The affected parent, the father, the O allele is associated with nail patella syndrome disease gene. We have these two recombination events. We can determine a recombination frequency. This recombination frequency can be determined from the 13 children that are from matings in which the genes encoding the nail patella syndrome and the ABO blood type segregate. 2 of them are recombinants. So to get to the 13, you basically have to take out, in the second generation, number 2 and number 6, because they married into the family, so they're not subject to this. The recombination frequency then, because we know there are 2 recombinants in generation 3, is 2 out of 13, which is approximately 15%. Pedigree analysis is still an important tool in genetics for mapping human genes. There are also additional, more modern methods that can support these studies, such as genome-wide association studies. In these studies, you identify a phenotype that is occurring in a large group of people. So this is based upon large numbers of people. Then you subdivide the populations into those that are affected and unaffected, and then you sequence the DNA from these individuals that are either affected or unaffected, and then you compare the sequences. And what you hope to do is identify SNPs, which are single nucleotide polymorphisms, and these become linked markers that can associate the genetic basis of the trait with a certain region of the chromosome. And of course, you have to do further analysis to verify which gene in that part of the chromosome may be causing the phenotype that you're interested in. So connecting a human gene to a phenotype involves a number of techniques. You can do family linkage studies and sort of determine how they're inherited, this particular illness, and you can potentially look for a mutation. You can do genome-wide association studies, find SNPs that associate with the phenotype, and look for a common variant among people who are affected. And then from these studies, you can create some hypothesis about what type of gene or what type of process this particular phenotype is involved in, because you get, especially with a genome-wide association study, you get a number of genes that surround the SNP that could possibly be linked to the phenotype. So then you take your candidate region and you can go back and sequence it in both the cases, which would be the affected people, and in controls, and see whether the cases, these affected individuals, have a mutation in a particular gene. And with the increasingly low cost of sequencing, this will make these types of studies a lot easier in the future, and we'll be able to link more and more genes to particular phenotypes. So I want to leave you with a few genetic terms and concepts. Linked genes are genes that do not sort independently. Recombination frequency is calculated by summing the number of recombinant progeny, dividing by the total number of progeny, and then multiplying by 100%. Interchromosomal recombination takes place among genes located on different chromosomes. Intrachromosomal recombination takes place among genes located on the same chromosome through crossing over, and this is what we've been focusing on today. Recombination rates can be used to determine the relative order of genes and distances between them on a chromosome. A genetic map is a map of the location of genes calculated using recombination frequencies. A physical map is a map of the location of genes based on their physical location on the chromosome. A map unit is determined by the recombination rate. One map unit equals a 1% recombination rate. Genome-wide association studies locate genes that affect particular traits by examining the non-random association of a trait with specific genetic markers, usually single nucleotide polymorphisms. And if you remember, a single nucleotide polymorphism is a single change in a base in DNA, in a gene, in a region that controls gene expression anywhere in the genome. So that's what a single nucleotide polymorphism is. I'm going to leave you today with an example of polyploidy. Here we're looking at a normal healthy human cell versus a cancer cell, and you can see that the cancer cell has a very different karyotype. And so next time we're going to talk about chromosome variation. See you then.